Hello students, in the last lecture we had talked about the non-cyclic photophosphorylation, and we had seen there are two photosystems, photosystem 1 and photosystem 2, which are involved in this entire procedure. Okay, now we are going to talk about something known as cyclic photophosphorylation. Okay, so we are going to talk about something known as cyclic photophosphorylation. Okay, we'll constantly be comparing cyclic photophosphorylation. Just a second. So we will be comparing cyclic photophosphorylation with the non-cyclic partner. Now remember one thing, in non-cyclic there are two photosystems involved. However, in cyclic photophosphorylation, for example, the phosphorylation which occurs in bacteria, the bacteria which are photosynthetic bacteria which are capable of production of ATP via cyclic photophosphorylation, in them there is only one photosystem, and that is photosystem 1 that is involved, which is this one, photosystem 1 that is involved. As we have seen, in presence of light, the light energy falls on this, the photons fall on this, and it gets excited. Similarly, over here, light falls on this and it gets excited. Well, that is okay. So once it gets excited, you have the primary electron acceptor who accepts the electron. As we had seen over here, you have the primary electron acceptor. Okay, and we had seen in non-cyclic photophosphorylation, from the primary electron acceptor the electrons move to ferredoxin. Actually over here it moves to ferredoxin and then it moves to NADP, which leads to the production of NADPH. But in case of cyclic photophosphorylation, the electrons don't move to ferredoxin. Rather, after moving to the primary electron acceptor, the electrons move to the plastoquinone. The electrons are excited and they don't move on this way. They move to plastoquinone, and from plastoquinone it moves to cytochrome B6, then it moves to cytochrome F, then it moves to plastocyanin, and then comes back over here, and then comes back. So the electrons are excited, and via this electron transport chain it comes back to photosystem 1. That is why it is known as cyclic photophosphorylation. Now why is it at all known as photophosphorylation? Because while the electrons are moving from cytochrome B6 to cytochrome F, of course there is this proton gradient that's created, which leads to the production of ATP. So in non-cyclic photophosphorylation, the primary electron acceptor donates the electron to ferredoxin and ultimately to NADP, which leads to the production of NADPH. But in cyclic photophosphorylation, NADPH is not produced, and photosystem 2 is also not involved, so water splitting also does not occur. So oxygen is also not released over here. I suppose this is clear to you. Okay, so in cyclic photophosphorylation, as I have seen over here, what happens is the electrons are excited, they move to the primary electron acceptor, and from the primary electron acceptor it moves to ferredoxin, and then it moves to plastoquinone, and then it moves to cytochrome B6, then it moves to cytochrome F, and then it moves to plastocyanin, and it comes back. So of course the whole, once the photosystem 1 gets excited, the electrons, they jump to the higher energy level, there is a hole that is created, and since the electrons are coming back over here, so photosystem 1 itself is involved in relaxing the whole electron hole that is being created over here. Okay, so the electrons are moving, they are getting excited, and then they are coming back. So it's a cyclic process, and hence it is known as cyclic photophosphorylation. Of course over here there is the production of ATP from ADP. So ATP is produced from ADP over here. Okay, so we see that definitely there is a production of ATP, but of course since from ferredoxin the electrons don't move to ferredoxin NADP oxidoreductase, and then it does not move to NADP. This does not happen over here. That is why that is why NADPH is not produced over here. NADPH is not produced over here, but definitely ATP is surely produced, and this type of photophosphorylation is known as cyclic photophosphorylation, where you have the ATP production but you do not have the NADPH production, where only one photosystem, that is photosystem 1, is involved, and ferredoxin does not pass the electrons to ferredoxin NADP oxidoreductase, rather it passes the electrons to plastoquinone, then to cytochrome B complex, then to plastocyanin, and that comes back to photosystem 1, and this occurs in case of bacteriochlorophyll.